Hi all, my name is Ronan Winkles and I'm a student at the University of St. Thomas in St. Paul, Minnesota, and I'll be briefly sharing with you today about my work on the photochemical fate of quinone outside inhibitors. So these quinone outside inhibitors are a class of fungicides that are very heavily used, actually up to 16,000 kilograms per county each year in the US. Yet little is actually known about their environmental fate at this point, and this is really urgently needed as they're suspected of having a lot of neurodegenerative effects on people that come into contact with them. So my research on these quinone outside inhibitors fits into a broader project in collaboration with the University of Iowa to assess the potential impact these fungicides may have on drinking water. So the first step to achieving this goal is to understand the environmental fate of these compounds before they actually reach that drinking water plant, looking at what sort of processes they might undergo as they're first applied to surfaces like crops, as fluoxystrobin is shown in the figure here, or to what happens as they run off into water supplies due to precipitation or irrigation, whether they make it there um, from there to the drinking water plant at all, or whether they might degrade into different compounds along the way. That might also be of interest. So the photochemistry of these compounds, that as they undergo it in the environment, can be roughly divided into two sections, aqueous and surface photolysis, as these compounds may actually react differently in the sunlight depending on whether they are sorbed to a solid surface or in solution. So we'll start with how they undergo photolysis in an aqueous environment. Um, so I'm presently looking at four different quinone outside inhibitors seen along the bottom of the slide: phenamidone, fluoxystrobin, diamoxystrobin, and cresoxim-methyl. They're each commonly used, but display some clear differences in structure, as you can see. And starting at the figure on the far left, we see that direct photolysis of these compounds. And this is run at 3 micromolar under simulated sunlight in ultra-pure water. So the compounds behave independent of the effects of some of the things that might be found in natural waters. And it can be seen that in this figure that some of the fungicides, like fluoxystrobin in orange there, absorb light at these environmentally relevant wavelengths and as such undergo significant direct photolysis. But others like phenamidone in blue hardly degrade at all actually under this light. And then cresoxim-methyl and diamoxystrobin are overlapping in the figure, but they also undergo some significant direct photolysis, though less than the fluoxystrobin. In further experiments we've done, as seen in the two figures on the right, each sample was then additionally spiked with 10 parts per million of nitrate and then nitrite, which are common components of fertilizers, but they're actually also known to incite indirect photolysis through the generation of reactive intermediates when irradiated. So this can clearly be seen through the increased degradation of all the fungicides tested. Even phenamidone, which is, as you saw in that figure on the left, doesn't undergo photolysis on its own. And from this, we know that the quinone outside inhibitors have the potential to degrade under aqueous conditions in agriculturally relevant areas. And additionally, all the detected photoproducts that came from this were more hydrophilic than the parent quinone outside inhibitors themselves, meaning that the products may be more likely to make it to the drinking water supplies. And further work can go into measuring the fraction of that parent compound that degrades as compared to the fraction that would make it to the drinking water plants. And eventually we might look into characterizing their photoproducts. On the other side of photolysis, that on surfaces, our work is still really ongoing, so I just want to share with you our methodology at the current moment. So first, we spot on these compounds onto a model surface and then evaporate off the solvent. And this is really important because we want to examine the compound as it is sorbed to the surface, not as it is in the solution. And then we irradiate it, just as with the aqueous samples, with environmentally relevant wavelengths from a solar simulating xenon arc lamp, and then after irradiation, each of the compounds are eluted from the surface with acetonitrile and then quantified in an HPLC.
One
So in
minute.
comparing this, thank you, to the aqueous experiments, we hope to establish whether there may be photochemical differences between these fungicides as they are in solution, for example, like in a body of runoff, to when they are sorbed to a surface, such as on a plant leaf or on soil. And then taking these two pieces of photochemistry together, we hope to build a more complete picture of what may be entering our water systems, and from that, better inform our efforts to improve the safety of our drinking water in the future. Thank you.